We will now do Week 6, Mendelian Inheritance, Part 1. The learning objectives for Week 6 are to give examples of conditions inherited in each of the Mendelian inheritance patterns, to discuss typical presentation, natural history, and risk of specific conditions, to analyze simple pedigrees for likely forms of inheritance and to determine risks to specific family members, to complete inheritance patterns problem set. Let's look at the exceptions to Mendel's law. Mendel's traits showed two distinct forms. Most genes do not exhibit simple inheritance. Genotypic ratios persist, but phenotypic ratios may vary due to outside the gene influences, which include multiple alleles, other nuclear genes, non-nuclear genes, gene linkage, and the environment. A genotype allele combination that causes death is by strict definition lethal. Death from a genetic disease can occur at any stage of development or life. In Tay-Sachs, the disease is lethal by age of 3 or 4, whereas Huntington disease may not be lethal until late middle age. In a population evolutionary sense, a lethal genotype has a more specific meaning. It causes death before the individual can reproduce, which prevents passage of mutations to the next generation. So if we look at this, we see that a lethal genotype causes death before the individual can reproduce, thereby removing an unexpected progeny class following a specific cross. And a double cross of a dominant allele may be lethal. An example of this is seen in achondroplastic dwarfism. Achondroplasia is the most common form of dwarfism. In fact, it occurs in about 70% of all individuals who have a form of dwarfism. The average height in males is 52 inches and in females 48 inches. This form of dwarfism is due to a mutation in the fibroblast growth factor type 3 receptor, the FGFR3 gene. This causes the premature conversion of the growth plate into bone by promoting the conversion of cartilage to bone, as takes place in the process called endochondral ossification. Most often, this is due to a spontaneous new mutation. It occurs about 80% of the time this way, from the father who is generally older, and this occurs during spermatogenesis. Homozygotes for this gene are presumed to be lethal, as they are in many other animals. If we look at the clinical presentation of someone with achondroplasia, you would find that there is an extra space between the middle and ring fingers, also called a trident hand. There's poor muscle tone and loose joints, frequent middle ear infections, which may lead to hearing loss, normal intelligence. There are delayed developmental milestones such as walking, which may occur between 18 to 24 months. Individuals with achondroplasia have shortened arms and legs, with the upper arms and thighs more shortened than the forearms and lower legs. They have a large head size with prominent forehead and a flattened nasal bridge. They have a curved lower spine, a condition also called lordosis or swayback, which may lead to kyphosis or the development of a small hump near their shoulders that usually goes away after the child begins walking. They have small vertebral canals, which may lead to spinal cord compression in adolescence. Occasionally, children with achondroplasia may die suddenly in infancy or early childhood in their sleep due to compression of the upper end of the spinal cord, which interferes with breathing. They also have bowed lower legs. This is a picture of a child that has achondroplasia, and as you see in this other picture, you can see the family of another child who has achondroplasia, and you can see that the family is of normal height and does not have the disease, nor does the elder sister. And you also can see that the father in this picture is a little older. These are two YouTube videos on the clinical manifestations of achondroplasia. In this slide, you can see if two people with achondroplasia have children, each child has a 2/3 chance of inheriting achondroplasia and 1/3 of being of normal height. This is a conditional probability because 1 in 4 conceptions does not survive. In incomplete dominance, what we have here is the heterozygous phenotype is intermediate between that of either homozygote. In complete dominance, only one allele is expressed and the other is not. An example of this is in familial hypercholesterolemia, an autosomal dominantly inherited disease, and the heterozygote has approximately half the number of receptors in the liver for the LDL cholesterol. The homozygous form for the mutant allele totally lacks a receptor, and so their serum cholesterol level is very high. Here we can see how the measurement of plasma cholesterol is reflected in 3 genotypes. The phenotypes parallel the number of receptors. Individuals with 2 mutant alleles die in childhood of heart attacks. Those with 1 mutant allele may suffer heart attacks in young adulthood, and those with 2 of the normal alleles do not develop this inherited form of heart disease. We can also see in this picture an artery that's blocked with cholesterol-rich plaque. Cholesterol is also deposited in joints and in many other body parts of individuals who have this disorder. Different alleles that are both expressed in a heterozygote are codominant. The ABO blood group system is based on the expression of codominant alleles. We see that the blood types are determined by the patterns of molecules on the surfaces of the red blood cells. Most of these molecules are proteins embedded in the plasma membrane with attached sugars that extend from the cell surface. The sugar is called an antigen, a molecule that the immune system recognizes and responds to. People who have blood group A have an allele that encodes an enzyme that adds a piece to a certain sugar attached to a plasma membrane, producing antigen A. Those individuals with blood group B, here we have the allele that is slightly different, and it produces the antigen B. People with AB have both antigen types, and those individuals who have type O blood actually don't have any. This slide is illustrating codominance. The ABO blood types are based on the antigens on the red blood cell surfaces, and this depiction greatly exaggerates the size of the A and B antigens, and the genotypes are in parenthesis. This slide is showing Punnett squares that show the genotypes that could result when a person with type A blood has children with a person with type B blood. Penetrance refers to the percentage of individuals who have a particular genotype who have the associated phenotype. Expressivity refers to variability in severity of a phenotype, or the extent to which a gene is expressed. The genotype is incompletely penetrant if some individuals do not express the phenotype, and the phenotype is variably expressive if symptoms vary in intensity among the different people. Pleiotropy is a phenomenon that exists where you have a single gene disease with many symptoms, or a gene that controls several functions or has more than one effect. Such conditions can be difficult to trace through families because people with different subsets of symptoms may appear actually to have different diseases. If we look at the disease Porphyria variegate, it affected several members of the European royal families, including King George III, and there were different illnesses and quirks that appeared to be different, unrelated disorders in these various people. In looking at porphyria, these are all diseases that result from deficiencies of any of several enzymes required to make heme. In each one of these diseases, an intermediate biochemical builds up. Sometimes they're excreted in the urine, or sometimes they accumulate in the tissues, and when they do this, they cause various symptoms. And some of these symptoms include reddish teeth, photosensitivity, and this is where we think that's possibly where we got the vampire and the werewolf legends. On a molecular level, pleiotropy occurs when a single protein affects different body parts, or participates in more than one biochemical reaction, or has different effects in different amounts. One of the diseases that fits into this category is Marfan syndrome, autosomal dominant disorder. The most common form is a defect in elastic connective protein called fibrillin, which is abundantly found in the lens of the eye, the aorta, in the humerus, radius, ulna, femur, fibula, tibia, phalanges, and ribs. This is Jonathan Larson, who died just before the opening of the Broadway play that he wrote, Rent, when his aorta ripped open. He had gone to 2 New York City hospitals complaining of chest pain, but no one recognized the symptoms of Marfan syndrome. Mr. Larson's long, thin face is characteristic of this disorder. The clinical manifestations of Marfan syndrome are a tall, thin build. Long arms and legs and fingers are spider-like, which is called arachnodactyly, and toes and flexible joints. A spine that curves to one side, this condition is called scoliosis. A chest that sinks in or sticks out, these conditions are called pectus excavatum or pectus carinatum. Teeth that are too crowded, and flat feet. In actuality, many of these individuals are on basketball teams, and it's unfortunate for that because one of the other signs we'll talk about in just a moment, or one of the other problems with this disorder, is the problem with the heart. In continuing on with the clinical manifestations, we see that they are prone to have an aortic dilation or aortic aneurysm. And today, if you catch it in time, you can put in a synthetic graft. They also can get mitral valve prolapse and lens dislocations. In looking at these pictures of individuals with Marfan syndrome, we can see a normal chest and we can see a Marfan syndrome chest of a man. Also, let's take a look at the arms. If you were to take your arms and put them to your sides so that you have like a wingspan, and you were to measure your wingspan, that should be the height that you are. However, if you have Marfan syndrome, their wingspan is more than their height. In the individual with Marfan syndrome, in comparison to a normal person, you can see how they have these elongated fingers, the arachnodactylia, and they also have these elongated arm bones, and they're able to wrap their hand completely around their wrist. In this picture of an individual with Marfan syndrome, you can see how they could put their thumb, how their thumb actually comes off the other side when they clench their fist. In this slide, the young man has been diagnosed with Marfan syndrome, and you can see his long fingers and his long arms, and he's a very tall boy. Please view this YouTube video on Marfan syndrome and the clinical manifestations of the disease, and you can also listen to some of the individuals who have Marfan's discuss their disease. The cancer genes are inherited in an autosomal dominant mode. And on this slide here, we can see the hereditary breast and ovarian cancers, HBOC. And we can see if individuals have those specific genes, how they have an increased risk of getting specific cancers, the breast, the pancreatic, the ovarian, and the prostate in comparison to the general population. And here are some of your HBOC syndrome genes, your BRCA1 and BRCA2, which were the first two that we knew about, the TP53, PTN, CDH1, ATM, CHEK2, and PALB2. In the past year, I have seen a lot more individuals with the PALB2 gene in the clinic that I work at in Fairfield County as a cancer genetic counselor. In this slide, you can see that multiple genes can increase the risk of a single cancer. We see that it's no longer just the BRCA1 and BRCA2 that can predispose you to breast cancer. We see all these other genes now that have been contributing to the cancer. And then we also see that multiple cancers can be associated with a single gene. When we get to talk about the different types of testing that we have for cancer, when we talk about panel tests, we'll see how important it is to be able to test in one test for many different genes. In looking at Lynch syndrome, we see that there are several genes which make up the Lynch syndrome genes, and these are individuals, if they have these genes, have an increased risk for colon, stomach, uterine, and ovarian cancer. Again, these are going to be inherited in an autosomal dominant form, and here again you see the general population risk with those. Here are the Lynch syndrome genes: MLH1, MSH2, MSH6, and PMS2. I would like to end this portion of this week's work by looking at a patient, Michael. Michael, a 16-year-old male, comes to the physician for evaluation of 2 episodes of nearly fainting in the last week while playing football. Michael has been on the varsity team for 2 years. His past medical history is unremarkable. A systems review reveals no obvious cause for his near syncope. However, a systolic murmur at the left sternal border is detected. A stat electrocardiogram shows ventricular hypertrophy, deep Q waves, and cardiac ischemia. A subsequent ECG shows a septum measuring 17 millimeters, consistent with a diagnosis of hypertrophic cardiomyopathy, which occurs every 2 per 1,000 in young adults. How could a family history and a pedigree help you to treat this patient? In reviewing the extended family history, you learn that Michael has an older sister, age 18, and a brother, age 20, both in good health. But you also learn that Michael's father died at age 39 in a car accident after a sudden heart attack at the wheel, and Michael's paternal first cousin died while playing tennis at the age of 15. This family history is suggestive of an autosomal dominant type of cardiomyopathy for which Michael's siblings and other family members are at risk. This information helps provide family members with a more accurate risk assessment and allows for medical intervention and careful monitoring of affected and at-risk relatives, such as Michael's 2 siblings. This slide shows you some of the autosomal dominant disorders that cause cardiovascular disease, such as hypertension, hypertrophic cardiomyopathy, and familial hypercholesterolemia. So we will stop here now for the autosomal dominant, and the next portion of this week we will be looking at autosomal recessive disorders.